Science is for everyone. Science is for everyone. Science is for everyone. C2ST. 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 Chicago Council on Science and Technology. C2ST. 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 Chicago Council on Science and Technology. Science is for everyone. Science is for everyone. Science is for everyone. C2ST. 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 Chicago Council on Science and Technology. C2.
Hello, everyone, and welcome. Thank you so much for joining tonight. My name is Kylie Kozlik, and my role at the Chicago Council on Science and Technology is to help put together programs just like this one for everyone to enjoy. So welcome to tonight's program, Proteins and Plants and Bees, Oh My, an assembly line for biology. This program is presented to you by C2ST in partnership with the Carl R. Woese Institute for Genomic Biology at the University of Illinois, Urbana-Champaign. Thank you for helping to bring this event to life by joining us live, and shout out to the Institute for Genomic Biology for help making this event possible. Now, if you are new to C2ST, we believe that science is for everyone. And what we do is create free science and technology programming for everyone to enjoy. If you want to learn more about us, check out our website at c2st.org for more content, more cool stuff. And if you want to support more events like this happening, feel free to donate by clicking the Give Now button on the website as well. Now, in a moment, I'm going to introduce tonight's guest, but before I do, we will have opportunities to answer your all's questions after the talk. So the QR code that you see up in the corner, that will take you to menti.com. If you enter code 2115-8394, you'll be able to enter, ask all your questions, and we will answer them at the end of tonight's program. The talk should last about 25-30 minutes, and then we'll move over to Q&A. We'll also have folks in the comments, so if you aren't able to access the Menti, you can also submit your questions in the comments of the video as well. So without any further ado, I will introduce our speaker for tonight, Stephan Lane. He is the manager of the iBioFab team at UIUC. He has a background in molecular and synthetic biology before moving into the amazing world of automation. So Stephan is going to kick us off by telling us a little bit about the work he and his team does. So why don't you go ahead and take it away?
Thank you very much for the introduction. So today I'm going to present a slideshow about some of the work that we do at our BioFoundry. So across a wide range of topics, from plants to honeybees, and even proteins. So a biofoundry is a robotic facility where molecular biology research is performed. And this allows scientists to do things at scales never before possible. And one of the first areas that these have made a huge impact is in the pharmaceutical industry, in drug screening and other applications. So these are some pictures of some commercial biofoundries at AstraZeneca and another pharmaceutical company. And the main driving point of setting these up is that reducing human intervention. So it'll allow scientists to spend their time focusing on solutions to large problems rather than doing small, repetitive tasks over and over. And so the first project I will tell you about today is our work in honeybees. So there are a few motivating reasons that we started this project. They are a scientifically important species. So these are studied as a model organism for a variety of purposes because they're relatively simple, but they have some very complex behaviors. They are used as a model for social interaction because they're a very highly social insect. And also evolutionary biology, genetics, and neuroscience all use these as a model organism. And addition to science, these have a lot of importance in agriculture as well. So they make products like honey that we are all familiar with, as well as beeswax and royal jelly, but they're also an important pollinator. So about 75% of the food crops that humans grow are pollinated by honeybees. And of those, about 80% are honeybees account for about 80% of pollination services of those. And overall, the crops that are pollinated by honeybees amount to about $14 billion. So it's a pretty substantial amount. But this has some problems associated with it. So how can we ensure that even when the climate is changing, that we can have a healthy population of honeybees continuously in the future? And a second problem is related to the application of pesticides. So in the past decade or so, there was implementation of a new pesticide, which really, along with some other factors, caused collapse of honeybee colonies worldwide. So we're hoping to develop a system that will ensure that this doesn't happen again in the future. So the solution to all these problems is that we wanted to develop a robotic platform where honeybees can be raised entirely in a controlled environment and also can be produced at scale. So this was the starting point of our research. This was a paper published shortly before we began our work into automation, and it is a miniature beehive. So this is made out of plastic, and you can see sort of a honeycomb shape on the object here. And so a queen bee will lay inside of this miniature beehive, and she will lay eggs into each one of these little honeycomb wells. And once the honeycomb plate is filled up with eggs, it can be removed, and then a scientist could raise those into adult bees in the laboratory. So we first worked on trying to automate the process that had been established for raising these honeybees using this type of plate system. And so this is an image of the plate, and this rectangular shape is sort of a standard for automating biology. So by having this shape of a plate, they were able to ensure that it would be compatible with automation systems. And so those little tiny lines in there are the honeybee eggs. And these are extremely fragile. So making sure that they didn't die was a major effort. And after a lot of laborious work and very tedious work, only a handful of these could survive. So we're hoping by employing automation that we could increase the scale of this and require less tedious handling by researchers. So these are some zoomed in photos of these honeybee eggs. So the way that we were able to hatch them and allow them to start growing was by providing them these tiny droplets of food. So on the left side there, you can see an egg with two droplets of the food placed near it. And in order for them to survive, we had to place these droplets with about a 0.5 millimeter away from them. And we had to feed as many as possible in a 20-minute time period. If we got the droplet too close, it would adhere to the egg and surround it, and it would drown the larvae. So it's a very fragile way to do this. But we thought that maybe by using automation, we could increase the speed of this and the reliability. So we started off trying to automate this process that they had established manually. So this is a picture of a plate with eggs in it. And we used a machine vision algorithm to find the eggs and return these feeding locations. And then these locations, which are shown in the yellow dots, were then fed to a robot, which would then place the droplets. And this is a plate that had been fed using the robotics. And there was some success on this, but in the first day, about half of the larvae died. So getting half of them to grow was still a success. But if we think about, there is about 10 days that we need to feed them. And if half are going to die every day, there's not going to be very many left at the very end. So we really tried to think of a new idea to make this work. And we came up with a new design for a plate. So I'd like to give credit to one of my colleagues named Adam Hamilton. This was a major breakthrough in this project, and this was his idea. So we came up with this plate design where instead of trying to feed the larvae from the top, we would allow food to flow to them by underneath, according to this diagram here. So we would only have to feed food on these wells that are along the side of the plate, and then it would flow underneath the plate. And by having sort of a mesh rather than a solid bottom, the larvae would be able to eat through it. And this is a image of the device that we designed to do the feeding. So there is a custom circuit board that we designed, as well as most of these parts were 3D printed. So this plate would fit into what we call a nest here, and it would move along a linear axis. And by having these two needles that were placed in a static location, they could hit all of the wells along the side of the plate. And we used a very high precision peristaltic pump. So this allowed us to get dispensing of liquids down to the microliter accuracy, which is what we needed. And here's a video of this in action. So there are two needles that are doing the feeding. And it takes less than a minute to do a full feeding. And it was hard to see the droplets in that one. So there's a little zoomed in photo, and you can see how the food is dispensed from the needles into the sides of the plate. And here is an example of some success. So on the far left, this is larvae after 4 days, and then after 7 days in the middle, and 10 days on the far right. So all the way from egg up until the final stage, which is this 10 days, we had pretty high success. And at this stage, 10 days, the plate would be sealed up. And then 12 days from then, they would emerge into fully formed adult honeybees. So now, this was working, but there was a lot of researchers manually moving plates between incubators to our feeder system. And we wanted to automate the whole process. So we designed a robotic system where this could be done that incorporated an incubator with temperature and humidity control, a robotic arm, and our custom feeder system. So we worked with a company who sells these incubators and robotic arms to design and construct this system. And the idea is that all of the plates would be housed in the incubator, which is along the left side, and then the robotic arm could grab a plate one at a time, move it to our feeder, do the feeding, and then send it back to the incubator. And overall, this takes only a couple of minutes. So that 20-minute window that we have to work with a plate at ambient temperature we are well within that limit. And so this is a video of the system in action. You can see the plate was just grabbed and sent to the feeder. They're usually equipped with a lid, and then the feeder system will provide food to them. And so the system, we set it up so that the user, all they need to do is set up the food for the honeybees every day. Once they set up the food and it's in a little temperature controlled environment, then they just press a button and the robotic system will carry out everything else. So each plate has a feeding log associated with it. So a researcher can go back and they can see how much food was provided to it at exactly what time and date. And there's also a full inventory of plates that would be generated at the end of a whole feeding session for a whole day. So you can see how many plates are in there, how successful they're doing, and how much food they are getting on every day. And this is an example of a plate that was fed using the fully automated system. This is at the final growth stage of a larvae. So after this, they will be sealed up and they will undergo something called pupation. And after 12 more days, they will emerge as adult honeybees. And overall, using this system to date, we have reared thousands of different plates, and our total success is about 60%. So there's still room for improvement. But if we think that initial system where manual researchers were getting maybe a dozen honeybees from a lot of manual work, this is a massive improvement. And the system we built can hold 504 plates, which means that at maximum capacity, this system can generate about 60,000 adult honeybees every 10 days. So looking forward, honeybees are very important, and developing a system that can scalably generate these is going to be very valuable. And our conception of the bee factory we see as one potential solution to this problem. And for example, citrus fruits require honeybees and other pollinators, so they will pay beekeepers to relocate their hives to their farm during the pollination season. But being able to produce honeybees at scale like this, we could provide them to different citrus farmers as needed. And additionally, protecting the natural honeybee population by using a system like ours to test the effects of different pesticides on honeybee larvae before they are deployed, and then the problems start to be noticed. And so while my efforts here mainly focused on automation and robotics, the person I mentioned who came up with the breakthrough of the plate, Adam Hamilton, he and Gene Robinson, who is the professor leading this project, have started a company called Forge Bee that aims to commercialize this technology and continue to develop it. So the next part I will talk about is our work on engineering plants. So plants are obviously critical to agriculture. They are a central part of it. And if we want to continue to produce more food in the future, we're going to need to engineer plants that have higher yields, are more resistant to pathogens, and can handle the effects of climate change. So from an idea to having a new engineered plant on the market requires about 10 years. And we are hoping that by using biofoundries, we can develop processes that will make that a lot faster. So using a pipeline that we developed, we aim to generate engineered plants that have increased oil production. So oil is important as an ingredient. It can also be used for biofuels and different bioproducts as well, but it's a valuable product of plants. So our general pipeline is shown here. We would first take plants and we would use their individual cells. They were called protoplasts, and then we can engineer these tiny cells. And if we see success at this tiny scale, then we can move the modifications into this middle part, which is called a callus. That's a mass of unorganized plant cells that will just continue to grow. And then on this larger scale, which this is already billions of different cells, if the manipulations to the genome are still providing the benefits, then we can move to generating full plants as shown on the right. The current method traditionally is people continuously work with these trying to engineer plants, which is slower. It requires a lot more space and a lot more handling. So by using this pipeline where we are optimizing the genetic modifications before we even get to a full-size plant, we are hoping researchers can study a lot more possibilities. And by the time they are generating these full plants, they already have been optimized. So here's a video of our biofoundry in action, generating these protoplasts. So on the bottom left is a video of the liquid manipulations that need to happen to generate these protoplasts. And the top right are some microscopy images of the individual plant cells. So we primarily worked with 3 different types of plants. N-benthamiana is also known as tobacco, which is a model plant. So starting with that is scientifically important. And then maize is corn, and sorghum is another important agricultural crop. And using this, we were able to screen for improving lipid production. And we were able to find a specific genetic manipulation that improved lipids between 2 and 4-fold. So next we moved to our callus, which is the unorganized cells. And we were able to modify them using an automated protocol and also confirm that for some of them, the improvements we saw at the small scale still held up, and they were still showing increased lipid production. And then after that, as you can see on the bottom right, these are full plants that were regenerated from the callus cultures. So this provides a full pipeline to get the whole plants after screening at a very small scale. And then lastly, one important thing to mention is that, as I mentioned, we were aiming to enhance oil production. And in the laboratory, detecting in an accurate way how much oil is being produced is something that is usually pretty slow and requires a lot of time and effort, and uses something called liquid chromatography mass spectrometry. And in order to develop a technique that was able to screen a lot more different constructs very quickly, we used a technique called MALDI. So this actually uses a laser beam. A single cell will be placed on a slide, and a laser beam will zap it. And then based on the types of basically dust that zapped cell emits, then we can tell what kind of lipids are in it. And using this, we were able to screen many hundreds of different samples very rapidly and at very low cost. And the data on the right is showing that our two methods are roughly equivalent, even though the new method is a lot faster and a lot cheaper. So here is a fluorescent microscopy image of the increased oil production that we observed in the plant cells. So the red is the cell wall, and the green are what we call lipid bodies. So these are just big globs of fats and oils that stick together because of their hydrophobic nature. So in addition to sort of an abstract measurement of lipids, you're actually able to see the increased lipid accumulation in our engineered plants. So overall, our effort, or our goal in this was not only to improve lipids, but to generate a proof of concept for using biofoundries to engineer plants faster and easier. And we are thinking that this started off with just a couple of plants, but every plant requires a new protocol to make it work properly. So we envision that this type of workflow will continue to be adapted for other plants that are also important agriculturally. So the last part I will talk about today is our work on proteins. So proteins are ubiquitous. They are very important for a number of industries. And for example, as shown here, biofuels, pharmaceuticals, even in foods and in diagnostics, research and development, they play a critical role. And in the 2024 Nobel Prize in Chemistry was awarded to a couple of researchers who were able to use artificial intelligence to predict protein folding. So the impact of this type of work is extremely massive. And our thought here is that by combining these new artificial intelligence tools for prediction with automation, that we'll be able to produce better proteins faster than ever before. So that concept is something we call a self-driving laboratory. This is where a robot is performing all of the experiments, whereas an artificial intelligence is generating the predictions. So they kind of flow nicely together. And this concept here has really taken off in material science recently, but its application in synthetic biology is very new. So this is the workflow that we developed. We would use, and it's circular, so it kind of feeds into itself. We would use an artificial intelligence to predict mutations of a protein. So since a single protein can have possibilities that are larger than the number of atoms in the universe. So a really complex model was needed to select these predictions accurately. Then we would use our robotic system to generate these mutants, as shown on the top right. And then we would do something we call protein expression. So these mutant DNA would be used to generate proteins. And then after we get the proteins, we would quantify how well they're performing. And then once we know which mutants are performing the best, we can feed this information back into the AI algorithm, and it would make more predictions. So everything feeds into itself and it's circular and it can keep going as long as we want it to. And for the predictions, we used 2 different models. One is called EV mutation, and this was produced, it's open source and it was made by an academic laboratory. And then the competitor is something called ESM2. So this was made by Meta, which is the parent company of Facebook, and they generated a large language model on protein sequences that works very similar to like ChatGPT that many of you might be familiar with. It just predicts likely mutations depending on the deep learning algorithm. So this pipeline we applied to a protein called phytase. So this is very important in the animal science industry. This catalyzes the reaction shown here. But in short, what it does is it takes some carbon, some nutrient that an animal normally can't digest, and it turns it into something that it can eat, it can get energy from. So this is energy that is normally locked up in animal food, but by applying this enzyme, they can get more nutrition value out of it. And so we aim to apply this enzyme to animal food, but one alternative is there is a company in Canada which created a pig which has phytase that it secretes in its salivary glands using genetic modification. So here are the results that we obtained. On the bottom, this is what we call a heat map of our results. The darker blue means worst performing, and the brighter yellow means it performed very well. So the very first predictions all generally performed pretty poorly, except for a handful. And then this information was fed back into the model, and each subsequent round, you can see how the predictions continue to improve. Until the final round, it was really giving a lot of high-performing mutants. And one thing I like to note is that ESM2, as this is the model that was trained by Meta, it did not perform as well as the open source academic model. And this is a sample size too small to say whether that's consistent, but I like to see, the academic scientists keeping up with the billion-dollar industries. So moving forward, we are hoping that this paradigm of combining robotics and AI to do autonomous experimentation will take off. And one thing I would like to note is that the models we used are some of the earliest ones that are available for protein folding. So they work very well, but we can expect better and better models to be released in the coming years. And our system, we can sort of mix and match these models. So as better models come out, we can implement them into our pipeline, and we can expect that our workflow will work even better. And this should have impacts across many industries, from biofuels and biochemicals to food ingredients, pharmaceuticals, and diagnostics. So a couple of take-home messages. One thing I like to note is sometimes people fear that robots will take our jobs, but I don't think that's the case. Rather than replacing people, it extends the reach of people. And I can give an example of this. When I was a student, I worked on microbial engineering, and I could publish a paper by engineering maybe 10 microbial strains. And in the age of robotics, and even in the future, 10 is just not going to cut it anymore, because all these high throughput techniques are being developed. So rather than replacing people, it is going to let them accomplish more. If I wanted to publish a paper today, I would probably need to produce closer to 100 different microbial strains. So the bar is being raised by people working with the robots. And another major thing is that people don't need to spend their time working on tedious manual tasks. They can spend their time analyzing data and thinking of big solutions to today's problems. Oh, I forgot to mention, this 3D rendering here is a new biofoundry that will be launching this year in July. And this will be aiming to provide synthetic biology as a service. So please note down my name. And if any lab or wherever you work wants to do some synthetic biology, you can come to us and you can contract the work out to us. Using our biofoundry, we'll produce proteins or microbes or plants for you. So I'd like to make a few acknowledgements. The Bee Factory was envisioned by Professor Gene Robinson, who's the director for the Institute for Genomic Biology, where I'm at right now, and Professor Huimin Zhao, who is my professor. And Dr. Adam Hamilton and Vasily Petrov had a major role in this project as well. For the automated plant engineering, this was led by Dr. Jia Dong, and as well as Huimin Zhao and Matthew Hudson were the professors in charge of this project. And the self-driving protein engineering also was a team effort with Professor Huimin Zhao at the lead and Dr. Nomani Singh, Jingxia Liu, and Tianhao Yu playing major roles as well. And we also have external collaborators at Thermo Fisher Scientific and my colleagues at ForgeBee, the startup company working on commercializing the bee factory. Thank you very much.
Awesome. Thank you. That was really, really interesting. We have a couple questions that have already come in, but if you are watching and you have questions, don't forget, there's the QR code up above. If you scan that QR code, it'll take you to menti.com and you can enter code 21258394. Also, you can throw questions in the chat and someone from C2ST will get them to me as well. So feel free, you can continue to add some questions. But the first question that we have here kind of builds on something you mentioned, which is, right, we have the AI that we have now, but how do you think advancements in AI technology could potentially impact your field in the future as AI continues to advance?
So like I mentioned with the robotics, not really replacing people, but raising the bar, that's how I see AI is going to impact things as well. That what we call an advancement today is not going to hold up 5 or 10 years from now. That an incremental advance in science will be a much larger magnitude than it is today. And AI is going to help with that.
Very cool. This is probably a quick answer, but another question that came in is kind of how many biofoundries are there? Is this like, are there a handful? Are there tons?
So we are a founding member of what we call the Global Biofoundry Alliance. This is a worldwide network of biofoundries. We were founded in 2014, and at that time there were only a handful. Today, there are about 150 members of the Global Biofoundry Alliance. The National Science Foundation this past year has funded the construction of 4 additional biofoundries, and there are a whole lot that are emerging in China. Korea also is investing close to $150 million in biofoundry development. So it is a rapidly growing community worldwide.
Awesome. And for folks who are watching who maybe are in college themselves or looking like maybe they want to go into a career graduating high school, like if someone wanted to go into the world of biofoundries and biofabrication, you know, where do you start? Is there a particular path? Do you even have to go to college? What's the kind of situation?
So biofoundry is a very applied field. There is electrical engineering involved, automation, and biology. So you'd really have to think about which type of, which part you're interested in. So for me, my background was in biology, and getting into biofoundries, that's still probably the way to go. But on our end, we don't work with software development as much, and the robotics, we work on programming them rather than developing them. If your interest lies in programming robotics, then go to school for studying robotics, or electrical engineering is probably the way to go.
Very cool. And then, you know, you mentioned 3 projects. I was just curious, like, do all these projects happen like one by one? Are you all like working on a bunch of projects at once? What does that look like? You know, is every day different?
So the first project was the bee factory, and that was chronologically also one of our first projects, one of my first projects. But the plant engineering one, there's a lot of troubleshooting that, and it took a long time. So that was a long project, and it overlapped with a lot of different sections. So I believe that project in some ways overlapped with every project I showed. And that is generally the way things go. Everything is a parallel process. There's usually one or two researchers that are designated a lead for a specific project, but we have to organize and share the robot time. It can get overwhelming with a lot of people wanting to use things simultaneously, but we have a calendar to make sure that doesn't happen.
Yeah, that totally makes sense. And, you know, a lot of times the questions we answer, you know, are because of where we're at or what's happening right now. But are there any kind of questions you have or research that, like, if there was the sky's the limit, you could study anything that you think would be really cool to explore with this technology?
That's a great question. I've never thought about that too much. I'm really interested in applying my work more to novel food ingredients. So I like rare sugars. This is something that has the potential to taste just as good as the sugar that we all put in tea and so on, but be healthy for you. So most of my work currently focuses on biochemicals and commodity chemicals, but switching to something that's more directed towards humans and direct to consumer is something I'm interested in. Most of our work currently, the end product would be something that is business to business. And the only way to compete is by making something that is lower cost than everything else. With business to consumer, you can kind of show the angle of, for example, there is a company that makes ice cream and dairy without cows, and it's more expensive than the natural ice cream, but no animals were involved, so no animals were harmed. And I like this type of angle where you can, it might be a little bit more expensive, but there are other intangible benefits that consumers might like. So I'd probably move in that direction if I could.
Very cool. We've got another question in from our audience, another couple. One is it common for organizations to use your BioFoundry to do research? What type of work do they do? And is data shared afterwards?
So we have had a few industrial partners, and that data is generally private because they are paying for it. Most of our collaborations happen within the university. So we do some work with the food science department. We're doing some work with the chemistry department. Especially one specific example is with the plant bioengineering work. The method I mentioned using a laser beam to quantify the oils, that was a brand new method developed by collaboration with the chemistry department. And so a lot of our work are large interdepartmental collaborations. And rather than just people generally currently don't come to us and ask, can you make 500 proteins for me? They usually come together as a collaboration where we all work together to publish a peer-reviewed article at the end of the project. But the change is coming in July. That's why we are building this new foundry, that it is designed so people can come to us and say, hey, we want 500 proteins, make them. And we can say, okay, here's how much it costs, submit the money order, and we will start building them for you. So this BioFoundry as a service is something that we are transitioning to in this year.
Cool. And so will you have like 2 labs at the same time, right? The one that people can do, and then the like research lab?
Yep, about 100 feet apart. Yep.
Very cool. Cool. Oh, this is an interesting question. So how long does it take for new research to reach, say, a regular consumer project, if it ever does?
That is a really good question. And the answer is always, it depends. But I will give a great example. So I work on a research effort currently called the Center for Advanced Bioenergy and Bioproducts Innovation, or CABI. And this organization started about 6 or 7 years ago. And the idea was that at the initiation of this work, which was funded by the Department of Energy, we would come up with a commodity chemical that is very close. It is just a little bit of microbial engineering away from being a reality. And the economics were there for it to be theoretically possible. So that was the idea we started with. And the first commercial product from that is about to be privatized. So I believe all the patents are filed. I won't say the product just so I don't get in trouble. I don't remember, but that is on the way. So it was about 7 years, but that was a massive effort between from computer modelers to strain engineering people to fermentation and scale up people. It was a massive team project to get that done in 6 or 7 years. Cool. So usually longer. That's a best case scenario.
Right. That's a project where like you had all the pieces in place, things were rolling the way they needed to. Very, very cool. Kind of, unless there are more questions that come in from our interwebs, which I'll give folks a chance by asking a question of my own. What would you say is one of the most like surprising or interesting things? Doesn't have to be the most, right? But a surprising or interesting thing that when you went into this work, you kind of realized or stumbled upon?
So when I first got into robotics and automation, I kind of had how do I say it? I hoped I could walk up to the robot, I could give it some samples, press a button, come back 3 hours later, and everything was done. The reality isn't that clean. Humans are still supporting almost every step of the process. And like I mentioned before, it extends my reach. I can get more done per hour than before, but I am still working in a different way all of that hour. Yeah.
Very cool.
That surprised me.
Yeah, that totally makes sense. So we did get another question in, which is, can you patent enzymes you develop? So like if you genetically modify a new enzyme, or if it's just simply yes, how does that work?
So this is a really good question because I had to consult our legal department on this. Typically, yes, you can. But my question came in when an AI algorithm is predicting different enzyme mutations. Does that change anything? Can we still patent it? And my understanding is yes. As long as those algorithms are being run on our hardware, and we're doing all the computation, then we can still patent it. But I wonder if that will change in the next decade. We'll see. As AI continues to advance, somebody might argue that AI-generated things shouldn't be patented. Let's see if it holds up. Because I know at one point, natural sequences were no longer patentable. And I wonder if this will go a similar way. We'll see. But right now, yes.
So yeah, right now you can. In the future, who knows? Very, very cool. Let me check, see if anything else has come in. Doesn't look like it has. So everybody, this is your chance. If you haven't asked a question yet, if something's been on your mind, submit it in the next, let's say, 2 minutes. And while we give folks that opportunity to think about that, are there any other kind of thoughts you wanted to add, or words for people who are excited by this science, or any places to keep out for updates, like to look out for?
So this general field would be called synthetic biology. If this interests you, that's where to look for grad school. Usually undergraduate students study molecular biology or chemical engineering, and then the synthetic biology comes in graduate school. But the work we do is all technically automated synthetic biology. The exception for that was the bee factory, which would be automated entomology. But if engineering proteins and plants interests you, then synthetic biology is the way to go.
All right. That sounds super cool. Let me really quick. I am going to check one other spot for any questions that may have come in. And then all right, it looks like we have one more question. Ooh, and this one's a little bit different. This is, how was it designing new equipment? And, at a relatively large scale. And how much does something, if you can share, how much does something like this cost?
So at the very end, I showed a 3D rendering of a new biofoundry that we are building. That is about $4 million in total, but that's really at the high end. Earlier, I showed the bee factory, which was an incubator, something we built, and a robotic arm. Altogether, that was probably closer to $225,000, which is still quite a bit. But this is one of the goals of the Forge Bee company, is to simplify and make that something like that a lot more affordable for academic scientists, for agriculture, and so on. As part of that, we developed a feeder device, which we built in-house. And that in total probably cost $1,000. It was very, very cheap. But once you get into commercializing something, there's a lot more of, we need to make sure that this is going to be reliable for 100,000 uses and so on. There's a lot more code. You have to meet standards that when we were building something in a laboratory, we didn't need as much. But I would say for a basic biofoundry that has synthetic biology as a goal, you're probably looking at $1.5 million minimum.
Thanks for sharing. Thank you so much for joining us today. It's been super amazing. It looks like we've got all of the questions that have come in answered. So yeah, any last remarks? I already asked you, but want to give you that chance.
Thank you very much for the invitation and the opportunity to speak. I really appreciate it, and I hope it was informative.
Yeah, it was to me. So on that note, if you would like to tell us what you thought of this program, did you think it was informative, interesting, exciting? Feel free to, in the Menti, there is a link to a survey that you can submit to tell us what you thought. C2ST will also throw that link into the chat. It's just a really quick survey, lets us know what's happening in your brain when you're watching, and what we should do more of. So please give us your feedback by filling out that survey. We appreciate it greatly. But yeah, thank you all for joining. Thank you to Stephen for sharing this awesome information and answering all of our questions. Shout out to our AV folks for making this stream happen. And also another shout out and thank you to the Institute for Genomic Biology at the University of Illinois, Urbana-Champaign, who helped bring this program to life. So yeah, fill out that survey if you want to tell us how you felt about the event. And go ahead, if you go to c2st.org, you can both donate to support future programs like this. There's also a place where you can sign up for our newsletter if you want to be the first to know when future programs are happening. So thank you all again. I hope you learned something new. And if you want to show your support in a simpler way, go ahead, like that video, subscribe to our YouTube channel, and comment what you thought. Thank you so much for joining us today. Have a great rest of your evening. See ya.